So before we actually get into the details of the cell cycle, first thing that I want to spend a little bit of time is going over why. So why do cells ever need to divide?
To make more cells.
To make more cells, why would you need to?
To keep going. Because mitosis happens.
Okay, so some cells are all constantly dying, so we need to regenerate, right? What else?
We get too big, so
Oh, that's a big problem. Cells can't just keep getting bigger and bigger and bigger, so we need to have them divide rather than each cell get bigger. So multi-celled organisms have the same size cells as smaller organisms. It's just that they have more of them. So to regenerate cells, to grow, what goes along with growth? To what?
Reproduce.
Ooh, perfect. That's not what I was thinking, but the other is to reproduce. We're going to talk about the reproductive stuff later, but we are going into the cell division stuff for today. And along with grow, we'd also be thinking about like differentiation. So grow and develop and specialization. So a ton of reasons to divide. Fun fact, I'm looking at you all now, and both metaphorically and very literally, you are all very different people than what I saw the first day of class. So back in August, what I saw, well, it was you, but every one of the skin cells that was on your face back then has long since sloughed off. And so now you are literally a completely different person, what I can see. Now your insides, a lot of the cells are the same, but your skin has completely regenerated since I met you. Where is it all gone? Dust. Blah. Okay, so cell division, super duper important to not die. Cool? Oh, and it's not just your skin. You have skin, kind of, all through your intestinal tract. Where do you think all of that goes? Yeah, the majority of your poop is your own cells. You're welcome for that fun fact. So with that, let's talk cell cycle and mitosis. Okay, so you should have this down. What process enables new cells to contain the full complement of the genome? Okay? What do I need to do before I can have a cell divide? What happens if one cell just splits in half? Why is that bad? They don't have what? You're close. What happens if I take one nucleus and split it in half? How much DNA is going to be in each half? Half as much. And then what if I divide again? Oh, this is a problem. So a general process that we can keep this nice and simple is we need to replicate the DNA, separate the DNA, and then split the cell apart. So I want to pretend that I've got a deck of cards. How many cards are in a deck? 52, right? Kind of similar to the number of chromosomes in our cells, 46. And if I photocopy each one of these cards, and now I have two full sets, right? And someone says to me, yo, can I play with some of those? And they're all shuffled up because they're just sitting inside of the cell. And if I say to you, sorry, I keep like choking. That's not what I would say to you. I say, here you go. Here is half. What do you think the odds are that this deck contains exactly 52 cards that are all correct? Okay, so the big focus is on how do we take this deck and split it so that both of the cells that are made at the end are identical to what we started with. Okay, so we're going to talk first about the cell cycle in general, but we're really going to hone in on that part called mitosis because of the importance that it has in making sure that you get a card, and that we're evenly splitting everything up. So to understand cell division, we have to go back to this word, chromosomes. I hate this word. It is used way too much in way too many overlapping contexts. A chromosome can be the condensed form of chromatin. A chromosome can be one piece of DNA. It gets icky. So we have a few terms that we're going to clarify here. What do you notice about bloop? Nope, that's not what I meant to do. Please hold. Go back. Go back. Oh my goodness. What do you notice about all the letters that are on this? First of all, what do the letters represent? Different, not nucleotides, but just different genes, right? So what do we notice about all of these? They match. What do we notice about all of these? They match, but they're a little bit different too, right? This has A, B, C, and D. This also has A, B, C, and D, but it's a little bit different. What do we notice about these ones here? They're straight up different. So we're going to start by looking at these. These are sister chromatids. The way I remember is the D is for doubled. If the DNA has been doubled, you have sister chromatids. Okay. And what you'll notice is in the middle they are held together. What is the area where the two sister chromatids are connected? The centro... Nope. Okay, look at this. More words that all sound way too similar. So chromosome, chromatid, we'll talk about all those different vocab terms. We're also going to have a lot of center something. This guy is the centromere. So the area where two sister chromatids are held together is the centromere. It doesn't need to be in the middle. It can be up here. It could be down here. It could be anywhere along the length of the sister chromatids, but it's always consistent. So like chromosome 1 always has its centromere in the same location. And of course, this is going to be a result of DNA replication that has occurred. Fantastic question. Let's come back to that when we look at the ones at the bottom. Okay. You know what? I'm just going to say it now. We'll keep it easy. And most of you guys have taken bio. So when I say version of genes, what word do I actually mean there? Starts with an A. Allele. Sister chromatids not only have all the same genes, but they're identical to each other. They have the same information. So, for example, if A is the gene for eye color, B is the gene for hair color. It's not this simple, but I'm way simplifying it. C is the gene for height, and D is the gene for weight. Then the version of one would be the same as the other one, same allele. So if this was the allele for brown eyes, then this would be the allele for brown eyes as well. If this was the allele for blonde hair, then this would be the allele for blonde hair, right? They're all identical, okay? Now I wish I was doing this in a different order, but we're going to quickly go over the ones in the middle. Those are obviously distinct chromosomes. They're completely different. They have different genes. And we say that these contribute to the organism's, quote, N number. So how many completely different chromosomes do humans have? How many completely different genes does... So we would say that the human N number is 23. So why do some people say we have 46 chromosomes? Because we have them paired. And that's going to bring us to this last grouping here. These pairs are called homologous chromosomes. They're going to have the same genes, but they could have different alleles. They could have different versions.
Is today your birthday?
Today? Yesterday was.
Yesterday was my dad's birthday.
Your dad has the best birthday.
I
didn't say anything?
Yes. Did you put that on the calendar like over there or something, or did you not say it? That's me. You're slick.
So.
Now we should also have.
Nope, I'm recording. You can't. So. After, there's already going to be so much music and festivities. It's going to be grand. I can't wait. Okay, where do you get homologous chromosomes from? So why do we have two pairs? Because we have two parents, right? And so we inherit different homologous chromosomes, or different versions of chromosomes from a parent. Yeah, we get a chromosome 1 from mom and a chromosome 1 from dad, but they could have different versions. Example, if this is the gene for brown eyes, what could this be? Maybe it's the gene for blue eyes, or maybe it's the gene for green eyes. I'm way oversimplifying, and that's okay. But it could potentially be different. Does it have to be different? Maybe mom gave you the gene for brown eyes, and dad also has brown eyes, and he gave you that gene. They could be the same, or they could be different, okay? Our focus today is really on the sister chromatids. We don't talk about homologous chromosomes until we get into meiosis. So exciting. Okay. And then the last thing I want to mention is that only diploid cells that have two sets of chromosomes, are going to have homologous chromosomes.
What's the like 2N?
2n refers to the fact that if n is 23, we have two sets of 23.
Oh, so it's like the pairs.
Exactly. Yep. And fun fact, I'll do this in red because strawberries. What do you think strawberries' number of different chromosomes are? They are actually 8, and they are octuploid, which is so fun. We're not that cool. If we were octuploid, that is incompatible with life, even having any extra chromosomes or too few for a full set is not compatible with life. Okay? So we've already seen this diagram. I'm just going to put it into context real quick. So this is in your chromosomal structures notes. I hopefully didn't change that too much. But this right here is a set of homologous chromosomes. This is chromosome 1, this is chromosome 1, but they're not identical. Okay? And how many different sets do we have? 22 plus the sex chromosome, so 23. Cool? Okay. So let's talk about the different types of cells. For the most part, in this unit, we're talking body cells. Sex cells are going to come later. What are the body cells called? Somatic cells. And what are the sex cells called? So sorry for what I'm about to do. So something that you should keep in mind if you are going into marketing or advertising, remember to always include gametes, because you know what they say, sex sells. Yep. That was good. That was so good. Did you say I'm going to use that one, or you're never going to? Okay, because if you were like, I am never going to use that one, that would have been heartbreaking. Cool. Okay. What process are going to make the somatic cells? Mitosis. So I remember this because it is from my head to mitosis. Right? All of those cells of your body are dividing using mitosis. What about for the sex cells? Those are going to use meiosis, and I remember that because the word meiosis has a sperm and egg built into the word. It does. Come on.
I don't see it.
It's
No, I see it.
Is that better?
Looks like a kite.
A kite? It's a sperm. It's a disappointing sperm, but that's okay. How many sets of DNA do your body cells have? A few. 2 sets. And so what do we call having 2 sets of chromosomes? What's that? Nice job. I've heard diploid. I say diploid, although I do understand that di would make more sense because di means 2, but I'm a diploid person that doesn't know any better. See what I did there? Okay, what do we call it when you only have 1 set of chromosomes? What did someone say?
I said
Unoloid. Okay, let's talk about why this makes sense. So imagine, if you will, that any old cell on my body and any old cell on my husband's body, they could come together, and those cells, when they came together, would make babies. Okay, we take a diploid cell, we take a diploid cell. And then magic happens, and what happens to my offspring? They are tetraploid, quadruploid. They are having way too many chromosomes, and what did I already say about having too many sets of chromosomes? That is not compatible with life. So the entire purpose of meiosis is to take the 2 sets and bring it back down to just 1 set. Same for the other partner, and the hibbitying and the jibbitying make a diploid organism. Good math, right? Because the 1 and then the, did we all follow that? Do you want me to write on the board? We can show how the 1 carries and the, it's 2. Cool. All right, any questions there? So let us get into the cell cycle. Here's what I want you to do. Put your pencils down. Do not write, just look up at the board. I'm going to walk through this whole thing in detail, walk through it again, and then leave the diagram on the board so that you can copy. Make sense? Okay. So the cell cycle just refers to what a cell is doing over the course of its life. Most of the time, it is in interphase. What cells do you think are dividing really, really rapidly, constantly? I'll give you a hint. Yeah. If I was to walk out of this room, I guarantee a bloodhound would be able to tell that I was here because just in the time that I've been here today, I have lost enough skin cells that it can be picked up by a bloodhound. That's crazy to think of. Running through the woods, you lose so much skin cells that a bloodhound is tracking you based on the dead skin cells that falls off, right? They're going through the cell cycle really, really fast. Interphase may only be 24 hours or 12 hours even. What cells are really not dividing fast, if at all? What types of cells do you think? What types of cells do you know don't really regenerate? What goes with that? What kind of cell would you have? No. Neurons and, what did I hear? That's kind of true. Muscle cells are also not going to divide very rapidly, if at all. In fact, if an adult gains muscle mass, it's not that they're making more cells, it's that the cells that are there are getting more fibrous and are just growing larger. So muscle cells and neurons, interphase may last 20, 30 years. It might spend the rest of your life in interphase and never go through the other phase again. So what is the other phase? We've got interphase, the time between divisions, and then the actual division, which is M phase. So let's start with the cell right at the beginning of interphase. The first thing that it's going to go through is what is called G1. That stands for gap, but you can think of G as what? Growth. So the very first thing that'll happen for a brand new baby cell is it'll grow a whole bunch. When it is time to actually divide, we're going to go through S phase, which means synthesis. Are we making more sets of chromosomes? We're just copying what we have, right? So the way I'm going to show this is that this cell becomes 2n times 2. It's got 2 copies of everything, okay? So G1 first, and then S for synthesis, meaning we are doing DNA replication. And then we go through G2. There's going to be a little bit of growth, but the big thing that happens in G2 is we make sure that that cell is healthy enough to actually divide. We give it a quick inspection, make sure that nothing's out of place, make sure there is no major problems so that we can go into the cell division phase. For the cell division phase, there's actually 2 parts. First, we have to separate the what? That careful card sorting that we've got to do. Make sure you get this and you get that. That chromosomal division, that careful sorting, that is going to be the nucleus actually dividing, what we call nuclear division, so that 2n times 2 will just become 2 separate diploid nuclei. So now we've got a cell with 2 diploid nuclei. What has to happen now? The rest of the cell, bloop, needs to divide, and that is going to be the process of cytokinesis. So I'm going to walk through the whole thing, and then I will leave this exact screen up on the board for a little bit so that you can escribe. Et voila. I don't even know what languages I'm going with anymore. We're just rolling through it. So interphase, M phase, we start here. We get some growing. We get some DNA replication going on. We double check to make sure we're ready to go. I really hate making that noise, but I'm gonna keep going. So we go through the nuclear division. And then finally we separate the rest of the cell. That is cytokinesis. And we're done. So our next job is to get into nuclear division. I can't wait. Do we feel okay about the cell cycle before we get zooming in on this little portion right here? Okay. Do you need another minute with this up? Boop. What's that? Yep, that leaves. That's exciting. We need to go to mitosis. Maybe. We're looking at the same EQ as before, nothing has changed, but now we're going into the process of mitosis, and to understand mitosis, we need to understand something called the mitotic spindle. You are going to hear me refer to this as both the mitotic spindle or spindle fibers. This is just part of the cytoskeleton, and in particular, it's the part that is responsible for moving and separating out the sister chromatids, okay? I want to correct this because for some reason it got cut off. Centrosome, centriole. We are going to use those two terms interchangeably, right? They're essentially going to be the same thing. And what they are like these little churro-looking things that grow the fibers that will then connect to and separate the sister chromatids, okay? So the location at which they attach to the sister chromatids, that is called this thing. What do you think that's called? Everyone says it. Kinetochore. The H is just there to feel fancy. So this is the kinetochore. It is what adheres the spindle fibers to the sister chromatids at the centromere. And what we're going to need are spindle fibers to attach at both ends. So we're going to walk through it. We're going to label some stuff. Then we're going to walk through it again. Then you're going to get tortured. We're going to watch some videos. So excited. Here we go. Let's start with a cell that is in interphase. Some of the characteristics of a cell that is in interphase. What color are we feeling today? Unfortunately, I can't do a dark green, so I feel like, is that too meh?
Pink.
I'm limited. We're gonna run with this pinkish purple. I got dark blue. I got medium blue. We're going purple. Okay, ADD. For starters, a cell in interphase is going to have very distinct DNA, or should I say very indistinct DNA. What do we say about DNA in this form? It's all loosey-goosey, noodly. I'm going to back off just a little and just call it chromatin. Maybe it's heterochromatin. Maybe it's euchromatin. There's some mixed in depending on what's being silenced. But it is definitively not condensed. It is not in the form of chromosomes yet. So it's all loosey-goosey chilling in the nucleus. What else do we visibly see around the nucleus? And I know it's kind of hard to see on yours, but do you see this wee little guy in here? Do we remember what that guy is? The nucleolus. One nucleolus or nucleolus. Multiple nucleoli or nucleoli. And somebody in first period was like, this might sound dumb, but what does the nucleolus do? And I was like, that is hella not dumb. How many people in here are like, I know what it does? Oh, see, not a dumb question. So when we talk about all of the DNA that is in the nucleus, it is responsible for being the template to make what type of molecule? So DNA acts as the template to make RNA. Right? What type of RNA does most of this DNA lead to the formation of? mRNA. The nucleolus is going to make rRNA, the ribosomal RNA, which is what makes ribosomes. So you would kind of be correct to say the nucleolus makes the ribosome, or at least part of it. Okay. And then these cute little churros up at the top. Centrosome. If you write centriole, are you wrong? No. Maybe eventually in college you might need to distinguish, but I don't know that I could even clearly tell you which one to use when, so we're fine. Okay. So you can tell that a cell is in interphase if these characteristics are there. Now, the rest of mitosis is like a beautiful dance. The pieces are moving around each other and swaying and separating and joining, and it's like a romance story gone awry or something. But it's very fluid. It's not like the chromosomes are like I'm here. I'm here. And so if I show you a picture and you're like, I can't tell if that's anaphase or metaphase, okay. But if you see a picture of two things and they're opposites and you say, that's, I will make sure whatever you see is clearly in a phase. And if you see something that's kind of in between, that's totally fine. You're going to see some pictures in between. Those are not going to be the ones that I grade you on. Okay? You'll see what I mean in a second. Okay, so let's start with early prophase. I know that this cell is no longer in interphase because one of the most obvious things happening is some condensing. So I start to be able to see sister chromosomes, sister chromatids. Okay. You see that our centrioles are starting to separate. And how, good job. My voice just cracked. That was exciting. How are they pushing each other apart? I don't know that this is super duper clear in your picture, and it may be worth drawing. Oh no, I did a bad thing. Please hold. Boop. Oh, all my writing is gone. Good. Boop, boop, centriole. Okay, so how are they moving apart? These little fibers that they are growing are going to start pushing on each other, moving them to opposite ends. So what do we call these? Either spindle fibers or the mitotic spindle. I like spindle fibers. I said that really weird. I'm sorry. Nope. Try that. Now we get into the end of prophase / kind of getting into metaphase and a phase that we'll call pro-metaphase or early metaphase. What do we notice about these centrioles? Where are they located? Ooh, love it. Perfect term. They're on opposite sides of the cell, which we're going to call the different poles. And each one of the centrioles is going to produce a spindle fiber that will eventually bind onto each of the sister chromatids. So for every pair of sister chromatids, it'll have a spindle fiber attached that come from each side of the cell. So the one that I have circled, you can clearly see that there's a spindle fiber coming from the one on the top and a spindle fiber coming from the one on the bottom. And again, what are the attachment points where the spindle fibers meet? The kinetochore. Nice. And so these are just going to push and pull and push and pull, and the kinetochore and the centromere is going to hold itself together really, really well until finally they will eventually give up. But for right now, they're holding on strong to each other. So as they are getting pulled and pushed by these spindle fibers, they're going to start getting lined up onto what will be called the metaphase plate. And as you'll notice, each one of these pairs of sister chromatids has a spindle fiber going in each direction. And so in this way, we're making sure that each cell gets exactly one full complement of chromosomes. If we've got 1, 2, 3, 4, 5, 6 chromosomes being shown here, we ensure that the cell that will become the piece at the bottom, that it gets one of these, 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 one of those. And that the one on top gets one of these, one of these, one of these, one of these, one of those, and one of those. Okay? So that now, during anaphase, what we're going to notice is that the spindle fibers are starting to shorten. And the way that they're shortening is that these kinetochores are kind of munching down, breaking them apart, and pulling the sister chromatids into opposite directions. And in fact, they're no longer sister chromatids. We are now going to call them chromosomes again. I forgot to mention one thing. We'll go back in just a sec. But where in the cell is all of this taking place? Trick question. Where in the cell is this whole thing taking place? It's basically filling up the whole cell, right? You'd have some like mitochondria kind of chilling in different places and some ER chilling just in some different places. But where is the nucleus gone? Yeah, we have to go all the way back to towards the end of prophase. Because what we see is that the nuclear envelope is completely dissolved by the time we get into metaphase. So there's no nucleus. They're just swimming around in the cell. Oh, that was too excited. Here we go. Then we get finally to, this is showing 2 processes in one. I just want to focus on telophase. During telophase, the chromosomes are starting to relax again. And what do we see starting to form? Okay. I'm going to write something on the side. It does not go specifically with this page, nor would I draw it if I were you. I'm just going to doodle something for a moment, and then we'll talk about why I doodled such a redonkulous thing. Okay, we don't need to make fun of the Doris hog. I don't know what that is. But what is it below? P on A. This will be sadly etched into your brain. The way you can remember the phases of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. The PP mat. And we can remember what happens in each stage with a little bit of a mnemonic. Remember to, first of all, always P before you do anything else. So PP goes first. Prophase, prometaphase. What happens during metaphase? Everything lines up at the... So if you see chromosomes in the middle, that is metaphase. If you see chromosomes that are starting to come apart, that's anaphase. And if you see 2 new nuclei, that was a really redundant phrase, 2 new nuclei. Nuclei. Anyway, if you see nuclei reformed and there's 2 of them, that tells you you are in telophase. What a cutie. Anyone want to guess why I never became an art teacher? We'll keep you. We'll keep you. Okay, and the very last piece, which we do see happening in this picture, is cytokinesis. Cytokinesis is when the actual cell splits apart. So after mitosis is most of the way done, the rest of the cell can now finally split. In animals, we're going to see this ring that forms around the cell that squeezes and makes a cleavage furrow. To cleave literally means to separate or to cut in half. It kind of looks like boobies. I'm sorry. It's just the fact of it. But when you see that happen, that is your cleavage furrow that will separate the 2 cells into their own daughter cells. Everything in biology tends to be female gendered. I don't know why. At least females get credit for something in science. And then on the plant cell side, are we just going to be able to squeeze from the outside? What do plant cells have? That cell wall. That makes a little bit of an issue. So the plant cell is going to have a whole bunch of cellulose just kind of dropped into the middle that starts to form a cell plate, then a cell wall. And that's how you're going to divide plant cells.
So how do plants get bigger?
Love that question. So wouldn't each plant cell be half the size as the parent? One of the main reasons that they grow is it's a little bit stretchy, right? When cells are first starting to form, the outside cell wall and the new one that they're forming are a little bit stretchy. And so they're going to have their vacuoles in the middle take up more and more water. And it's going to be that turgor pressure that allows them to grow. I love that. Okay. What questions have we got? Would we like pain first and end on a better note? Or start out good and then cause pain?
We still have to sing happy birthday.
Oh, no, we don't. Sorry, I'm still recording. Okay.